Hola a todos, bienvenidos a los seminarios institucionales del Instituto de Biología de la UNAM. Primero que nada, quiero invitarlos a todos a que se suscriban a nuestro canal y que le den clic a la campanita para que les manden las notificaciones de todos los seminarios nuevos que haya. Tenemos uno cada semana y hay veces que hay extraordinarios. Ahora queremos dejarlos con la doctora Patricia Ornelas, que nos va a presentar a nuestra invitada de hoy, Suzanne McGaugh.
Muchas gracias, Ale. Gracias también a los organizadores por permitirnos este seminario, y le doy las gracias también a la doctora Susan. Voy a hacer la presentación en inglés para que ella pueda saber parte de lo que estamos diciendo, aunque sé que ella habla un poquito de español, pero bueno, para poder. So, I'm pleased to present you Dr. Suzanne McGaugh, who is currently an associate professor at the Department of Ecology, Evolution, and Behavior at the University of Minnesota. And just a brief summary about her trajectory. Dr. McGaugh got her bachelor's degree in 2003 at the University of Texas at Austin, and her PhD in genetics in 2009 by the Iowa State University. She has an outstanding professional experience. So after her PhD in 2009, she got a postdoc as a research associate at Duke University, and between 2013 and 2014, she got a postdoc associate fellowship at Washington University. In the spring of 2014, she got a postdoctoral research associate at the Iowa State University, and shortly after, she got an assistant professor position at the University of Minnesota. Her outstanding trajectory has been recognized by honors and awards, and in 2009, she got the Iowa State University Research Excellence Award, and in 2010, she was honored with the Outstanding Postdoc Award by the Duke University. With a total of 44 articles published, 1 book edited, and 2 book chapters, she has published her studies in high-impact journals like Nature Communications, Proceedings of National Science, Academy of Sciences, and among other high-impact journals. Dr. McGaugh is the principal investigator and a collaborator of 4 active projects funded by National Science Foundation and NIH. Her research interests are focused in 2 main topics. The first is how fast organisms can adapt to new environments. For this topic, she investigates genomes for signatures left by local adaptations, particularly working in cavefish from Mexico and other model organisms. And the second main topic is related to how recombination shapes genomes. She is interested in the causes and the implications of recombination in different model species. It is a real pleasure to having you, Susan, in our institutional seminar. I really thank you for accepting to be here today, and I will give you the voice for your presentation. Thank you.
Thank you, and thank you so much to the organizers, and thank you so much to Patty for the invitation. It's really a pleasure to speak today. And I'm really excited to share with you what my lab's been doing for the past few years, and much of it's been in close collaboration with Patty. I just wanted to start with saying it's a really amazing time to be an evolutionary biologist. You know, we have these classic model systems, and we understand so much about the evolution and natural selection in these systems, but now we're really starting to understand the genetic basis of these textbook evolutionary examples. And so it's a very exciting time, because we probably wouldn't have realized that this was the difference in coloration in the classic moth system was due to a tandemly repeated, a transposable element that happens to impact a cell cycle gene, or that integration was really responsible for the shape of pointy beaks in the Galapagos finches. And so there's actually a haplotype that bounces around between different populations and causes birds to have the more pointy beak. And finally, in our classic example of stickleback fish with their spines, we now know too that there are fragile sites within the genome that happen to break more often than normal in replication. And these fragile sites lead to the loss of the enhancer that is responsible for the PEDX1 expression. So my lab is incredibly interested in understanding what are the commonalities of these evolutionary molecular paths to phenotypic change, and can we explain and potentially predict characteristics of adaptive substitutions. We do this in 3 ways in my lab. So the first is that we look at pathway evolution. And we oftentimes look really deep in time for that. And we try to understand if there's certain genes that have sped up across large swaths of evolutionary history, or maybe that they show positive selection across equally large swaths of evolutionary history. We also look at genome content variation. So for instance, are there differences in the number of genes, or are there differences in the amount of material that, presence-absence variation that we see for genes among different species? And finally, what I'll talk to you about today is repeated evolution. And these are cases that we have extreme power to be able to understand natural selection and the genetic basis of phenotypic change. And one of the classic examples for this is in the garter snake in California. And so this particular garter snake is really good at withstanding tetrodotoxin. And so they can consume amphibians that contain vastly toxic amounts of tetrodotoxin. Now, this hasn't just happened once throughout evolutionary history. This has happened multiple times across different snake lineages. So within the colored branches, you see different evolutionary origins of this ability to consume tetrodotoxin-bearing amphibians. What's unique about this is that despite it happening over a large portion of the tree for snakes, we see that these changes are happening basically in the same regions of the same protein. Now, that's remarkable in itself, just among the lineages of snakes, but it's even more remarkable when we consider that evolution has acted on the same protein, largely on the same sites, even when you're talking about tetrodotoxin resistance in pufferfish. So what's cool about this, and what's really powerful, is that we know that mutational screens in the lab can create phenotypes or phenocopies using different genetic mutations. So there's lots of ways to get these phenotypes if we create these mutations in the lab. But in the wild, we often see similar mutations and in the similar genes. So this has been shown for Arabidopsis flowering time and Drosophila bristle number. We can recreate these phenotypes with lots of different mutations in the lab, but we only ever see nature take a few paths. So our lab exists to be able to try to understand what are those governing principles of those few different mutational spectra that are occurring. So there are three main things that pop out in the literature about repeated evolution. So these are the guiding principles. Oftentimes, they're genes that are affecting phenotypic change. They only affect the phenotype of interest, and they don't have large deleterious consequences. So pleiotropic consequences across the body are the phenotypes that were off target. Alternatively, there could be potentially that there is a different genomic context. So regions of high recombination rate oftentimes evolve quicker because they are able to dissociate themselves from bad regions of the genome and be able to proceed during adaptive evolution much faster. Likewise, we talked about fragile sites being places in the genome that break more often than others. So finally, it's also the case that you could just have a really big gene, right? And that often happens. So where a really big gene is just more likely to get hit by mutations than a really short gene. And sometimes those mutations can lead to phenotypic change. And that's the case, or potentially the case, in this example I'm showing you from cavefish. So you probably have seen more than you want to know about osteonyx by Patty's work, but this is the surface version of Osteonyx mexicanus. And here are two different cave populations, Molino and Pachon caves. Now, this gene is huge. It causes albinism across many different vertebrate species, but it's 120 kb long. So that's a huge amount of regions for a potential mutation to be able to land. And in fact, what we see is that exon 21 is deleted in Molino cavefish, and exon 24 is deleted in Pachon cavefish. So these are independent evolutionary origins causing a similar phenotype, using the same gene, but deleting different parts of that genome, that particular gene. So this is illustrative of the power of the cavefish system in looking at repeated evolution and what we can learn about the evolutionary process. We have 5 main caves that we work on in the lab: Molino, Pachon, Los Sabinos, and Tanaja and Chica. And again, all of this work is done in close collaboration with Patty. We will use, for this talk, I'll use 3 main caves, Molino, Pachon, and Tanaja, the blue, red, and green. So there's 2 lineages of surface fish within this that gave rise to each of them gave rise to at least 1 cave colonization. So we know we have at least 2 independent evolutionary origins of this cave phenotype. And if you haven't seen these in action, we're just going to watch a quick video here. So they're very good at swimming around the tank. And despite not having eyes, they have this expanded lateral line system that helps them detect vibrations in the water, as well as where they are relative to each other and relative to the rest of the tank. They're also incredibly good at detecting food. So the caves are a very nutrient poor environment. And so they seem to be very good at knowing whenever food is available, either through detecting vibrations or also having a heightened sense of smell and taste. So we have these tribomorphic syndromes, right? They're retinal degeneration, they have pigmentation reduction, they have expanded cells that help them detect where they are in the water. They have enhanced taste buds, this vibrational response that you saw just a moment where they swam towards where the food just dropped in. They also have differences in sleep. So cavefish sleep considerably less than surface fish do, and we're still trying to figure out why that is. And they have largely disrupted circadian systems. Finally, they lose weight a lot less quickly than surface fish do, and they gain weight much more quickly too. So in 2014, we published a short read genome in Nature Communications, and so we have also in press right now. It's accepted, but still in revision, the surface fish genome. So we have these genomic tools to be able to start to look at the genetic basis of these traits and the repeated evolution of these traits. So I'm going to tell you 2 different stories. One is the evolution of sleep loss, but we're going to cover the population genomics of this system first. We need to know some basic information about the cavefish, right? We need to know, are there really actually 2 independent origins of cavefish? Is there gene flow between cave and surface and between different caves? And how old are the cave populations? We always get asked too, like, how big are the cave populations compared to the surface populations? And can selection really drive these traits, or is it all just disuse? And so we started out with 3 different cave populations and 2 surface populations. So the caves are in blue and the surface populations are in white. We also sampled, so this is Osteonyx mexicanus, their range, and then this is Osteonyx igneus, and which has served as an outgroup. So we sequenced an outgroup as well. So we started this project in 2013, and so it's a little bit weak on the sampling, and we have since expanded it, but this particular study I'm going to talk to you about had 47 samples, sequenced at pretty low to moderate coverage. And again, we did this whole genome sequencing data. What we found out is that there's 2 lineages of cavefish. So we see that, there's a surface fish that's a sister taxa to a cavefish. Same here, we have the surface fish that's sister taxa to these cavefish. And so we can say that, yes, indeed, we have lineage 1, well, I'll just call that the red lineage, lineage 2, the blue lineage. And this was in collaboration with Uni Brandvain and postdoc Adam Herman. So this was generated with that whole genome sequencing data. Now, we also want to know how much gene flow is affecting the cavefish, because surface fish do get into the caves. But the question is, do they last long enough to be able to spawn? And so we have this, surface fish that's been swept into a cave in the last rains, and that's not a normal behavior for a surface fish, right? Swimming on your side and being covered in lesions is not really the healthiest behavior in the world. And so, so this guy is pretty much on his way to becoming cavefish lunch, and we want to know, do they survive long enough to spawn? To do that, we looked at an admixture plot, and what you're seeing here is each individual is represented by these bars, right? And so we have our red lineage and we have the blue lineage, and that's great. They're separating out beautifully, but we do see that we have some flow from the blue lineage into the red lineage, from the red lineage into the blue lineage. And so we do know that there's some cave to surface, and even surface to cave gene flow. Now, this is contemporary gene flow for these admixture plots. It usually picks up things that are pretty recent. So if something like this looks like it could be maybe a backcross, right? So this is the proportion of ancestry from the dark blue, proportion of ancestry from the light blue. And so that looks like it's about 25% dark blue and 75% light blue. And that's, so this is picking up really recent gene flow. We can also use a tool called TreeMix and be able to ask about more deep in time gene flow. And we again see that also in historical gene flow, there are substantial amounts. Now, this is important because it helps us understand more about repeated evolution. If it was truly independent origins and truly independent everything, we would see these de novo mutations that don't get transferred between the caves. But in fact, what we see is that there's a huge opportunity for gene flow to affect adaptation in the caves. And one of those examples is best illustrated by a QTL map. So these are places in the genome, these little gray boxes are places in the genome where we've mapped different traits to the genome. So these are all different chromosomes, right? And so we've mapped different traits, and you'll see that some of them are clustered. So this particular region right here is where eye size is being mapped to the genome. However, there's lots of other traits that are related to cavefish that are also mapping to that region. What's interesting here is that particular region shows evidence of being transferred between different caves. So it has really, really, really low divergence between the different cave populations, but totally normal surface-to-surface divergence. And so the only explanation for that is that gene flow is actually moving this little region in the genome across the different caves, and it's important for cave adaptation. Okay, so this is important because it helps us understand, okay, are we really going to be able to count these examples as truly independent origins and be able to learn quite a bit about the evolutionary process by looking at these different caves? Or are they kind of pseudo-replicates in that it evolved once, the evolution of the caves evolved once, and then just kind of transferred among the different caves through gene flow? So we modeled this using DADDY, and this was work by Tom Kono. And so what we see here is that we have an ancestral population. So that's what the NA stands for. This is how these models are set up. And they split into daughter population 1 and daughter population 2. And they do so at time TS, okay? So we have this ancestral split, and this is all modeled with DADDY. Now you can add on to these basic models. So this is a strict isolation model, and we're trying to see which of these models fit our data best. And we added a couple other examples and scenarios. So one is a isolation migration. So there's constant gene flow. Another example that we modeled was ancestral migration. So we had gene flow and then a time period where that gene flow stopped. And then finally, we had a secondary contact model. And we thought that the secondary contact model would probably best represent cavefish, where you have this ancestral population of cave and surface, right? Like before they divided into caves, before they evolved into cavefish. And they separate due to some kind of event. And so that we have a surface fish lineage going off this way and a cavefish lineage going off this way. And maybe occasionally after this period of divergence, they come back into secondary contact. So this is the model we were favoring, but we also added another layer onto that model in that we wanted to allow for parts of the genome to be protected from gene flow. These could be regions of the genome that are highly adapted to cave environments, and then by selection, they act to be protected from gene flow. So we added 2 different rates here. So that's represented by the green arrows and the black arrows. By and large, what Tom found is that when we modeled our caves versus our surface, we found evidence that this model fit the absolute best. So that we have this period of divergence between the cave and surface fish. They were isolated for some amount of time, and then they come back together and have some gene flow. And then there's parts of the genome that are easily interchangeable between cave and surface fish, and there's parts of the genome that are not easily interchangeable between the cave and surface fish. So we have a model for how the demographics work. So what Tom did is he took our tree and he was able to put some dates on this through our DADDY modeling. And what we found is that the pink lineage and the blue lineage separated from one another about 250,000 generations ago. And both lineages of cavefish about the same time came about 160 to 190,000 generations ago. So this is putting us a little bit closer to present day than what previous estimates had. But there's been a lot of debate within the cavefish community about how early these guys evolved and how recent they are. We also know from Tom's work that we can start to put bounds on this gene flow rates between different cave and surface, and that's represented by the different thickness of the arrows. So what I'm showing you here are simply just the gene flows across the different cave populations, because it would get really messy if I tried to do that between cave and surface. But what we see is that we are getting, somewhere between 2 and 3 migrant chromosomes, or about 1 and 1.5 individuals per generation on the thickest arrow. So there's a lot of gene flow. Okay, so what I've shown you so far is that the cave lineages are actually pretty young, as far as like the origin of cavefish. They're exchanging genes a whole lot with the surface fish, and we know that they have higher effective population sizes than what we really thought before. So what I'm showing you here is effective population size on the y-axis and the different surface populations here and the different cave populations here. So when we look at this, the surface fish actually have really high effective population sizes. They are like the Drosophila of the fish world. So we have zebrafish level, or Drosophila level, fruit fly level, Ioannese. So they're highly diverse. The cavefish, of course, are, as you would expect, much less have a much smaller effective population size, and that's fine, right? We were expecting that. What we weren't expecting is that they would have effective population sizes similar to humans. So, for instance, Molino cavefish, even though they are the most inbred or the most homozygous population we have, they actually still have, effective population size sort of similar to humans. Tanaha is our most diverse, and they have 3 times that amount. So what does this tell us? Well, when we go back to the literature, what we see is these explanations for the loss of eyes and for these cavefish traits that are based on this idea of disuse. Like the cavefish had no use for them, drift acted over time, completely obliterated these phenotypes through mutation. And that's been echoed not only by Darwin, but also in recent literature as well. So we have these examples where, they have lost their pigmentation in eyes following relaxed selection. So that really just doesn't make sense with what we understand about the biology, right? And that is that it is expensive to maintain eyes whenever you don't need them. And it is expensive to maintain a circadian rhythm when you don't need it. And caves are very low in nutrient input. So if you're in an environment where you are trying to conserve your energy the best you can, getting rid of your eyes makes a lot of evolutionary sense. Now, if that's the case, we're expecting selection to actually be acting to get rid of these traits. Now, with what I've shown you is that the caves are much younger. They exchange a lot of gene flow with the different cave populations and with surface populations, and they have effective population sizes that are a lot higher than we thought. Well, what does that mean? Altogether, we can say that there could be signatures of selection still left to detect, and that selection could probably easily act on these traits. So what we did is we took all of these parameters and we contacted Cartwright, who had already developed a model of evolution for eyes. And so he said, okay, we think that there's 12 loci, 12 genes that are involved in eye phenotypes, because that's how many QTL have been mapped to the genome. So we have 12 different locations. So I'm going to model something with 12 genes impacting eye loss. And we're going to use the time-sense divergence of cave and surface fish, the migration rates we modeled between cave and surface fish. And we're also going to model that secondary contact between cave and surface fish. And we're going to use the effective population sizes that we estimated, and we're going to put all that in Reed's model and estimate whether or not their selection coefficients are out of bounds of what we typically see in natural populations for selective sweeps. So what I'm showing you here, and I'll play this video in just a second, but I wanted you to orient to this first, is that what we're seeing here is that in blue, we have very high frequency of cavefish alleles. So this would be a cavefish allele, at 100%. And this would be surface fish alleles at 100%. So the red would be 100% surface fish alleles. And so what we're going to watch here is the selection coefficient here on the y-axis, and we're going to watch it drop down as we go. And so as you see, the selection coefficient needs to get, less and less as time goes by to be able to maintain cavefish alleles and to be able to select on these cavefish alleles. And so whenever our whole span of time that we modeled is over, what you see is the selection coefficient to drive cavefish alleles to fixation needs to be about 0.01, but between 0.01 and 0.1. So this isn't crazy high. This is the same case as what got rid of this pelvic spine in freshwater stickleback in about 8,000 generations. So this is like totally within the realm of what natural selection can do in a population. And so with that, we conclude that it's very likely that selection is driving these alleles for blindness to high frequency. So to conclude on this part of the talk, I wanted to just recap. We found that there's multiple lineages of the cavefish origin, although there is gene flow between the populations, so we need to be really careful about how we talk about repeated evolution, whether or not it's de novo origins of mutations, or if it's actually one origin and then spread through gene flow. And then in addition, they're much younger than we expected. These cave populations haven't been around as long as we thought. They have much higher NE, which gives them a much higher likelihood of being fixed by selection. I also wanted to just touch on one kind of public service announcement. So one kind of cautionary tale, and that's that relative measures of divergence can really be misleading. So this has been building in the literature for a while, probably for the last decade. People have been talking about how FST can really lead you astray. And so with the more frequent use of RNASeq data, I wanted to touch on this too, because I think it's a really important point that our data illustrate extremely well. So an absolute measure of divergence, so relative measures of divergence are going to be something like FST. An absolute measure of divergence is something like pi or dxy. And the way that we calculate this is that we total up the total number of sites. So let's say this is one chromosome and this is another chromosome from the same population. And let's say there's 100 different sites along this chromosome. And I can count up the differences between the different individuals. And so there's 7 differences, and I just say 7 over 100. So it's a 7% pi. Now with dxy, I can do the same thing, and dxy is between species, so we always expect it to be higher. It's represented by having a different colored line here. And so with this, we have, a dxy of 10%. So this is probably one of the better ways to measure divergence. And the reason is, whenever we're looking at FST and pairwise FST between different populations, there's an assumption with pairwise FST comparisons that you're comparing apples to apples. You're not comparing a population that has very, very low diversity to one that has very, very high diversity. If you're doing that and you're not taking into account the low diversity, that can artificially inflate your FST. And I'll show you a little bit more about that in the next couple slides. So what we're showing here in this example is the pi. So that's going to be the diversity among the population. So this is how diverse each population is whenever you're looking at the diagonal. And I only want you to worry about the diagonal for this. So this is Tanaha population to itself. And then this would be a surface population to itself. So you can see that in this population, there is more green. It's super diverse. You can pick out an individual in the surface population, and it's going to be really different from the next individual you pick out from the same population. Alternatively, for something like Merino population, which is a highly inbred, very homozygous population, it's dark, dark red, meaning that it has very little diversity at all. So whenever cavefish were first being looked at, they were mainly being looked at through FST. And it's a big no-no to do pairwise FST when you have this kind of situation, where you have this much heterogeneity in the diversity measures. So the reason for that is when we look at FST, you can basically break down FST into dxy minus average pi over dxy. So this average pi is really where it gets tricky and really where it can lead you astray. So if average pi is super low, so let's say I'm comparing 2 cave populations, and this average pi is going to be super, super low. So let's push that down. FST is going to get artificially inflated. What that looks like is that, so here I have pi, and then I have a metric of differentiation. So this is average pi between the 2 different populations I'm comparing. So what I want you to first think about is FST. So these two are cave to cave. FST is super high, right? Average pi is super low. For surface to surface, FST is super low, but pi is super, super high because they're both really diverse. Surface populations are a lot more diverse than cave populations. What we see when we use the absolute divergence is that this is wrong. And this has led us astray in the cavefish community for a long time, and also in other population genetic studies. So for a long time, we thought, oh, these caves are super divergent from one another. They must be really deep in time. They must not have much gene flow between one another. These service populations, on the other hand, they are super close to one another. They're super alike. They must all be having gene flow and be one big panmictic population. But that's all driven by this pairwise pi. And so it's really scary for people who are doing a lot of FST work, right? Because when we look at our absolute divergence, and we can do this because we had the genome sequence, because you have to have the invariant and the variant sites. You have to have the polymorphic and the not polymorphic sites to be able to calculate the absolute divergence, because it's out of the total number of sites you're looking at. And so when we look at this with DXY, what we see is that DXY between caves is actually even a little bit less than the DXY between surface populations. So we're seeing this thing that totally led us astray through using pairwise FST, and we're getting a much better capture of what the diversity and the divergence are of these populations when we use whole genome data. Okay, that was a lot, but we're redoing this work so we've started, this was the original study, was 47 samples. We now have 257 genomes representing 18 caves and 9 surface locations. All of the sampling was done by Patty's group. And for that, we are forever grateful for their help and for providing the basis of this study. And this work, the computational work, is being done by Rachel Moran and Daniel Drabeck, 2 postdocs in my lab. And so I'm hoping to come back and be able to give you a much broader picture of how many origins are and how much gene flow it is and how much that gene flow is affecting adaptation. And so we're hoping to do that. Really, we're in the process of doing that right now. So one key thing about this is that, I talked to you about how we can have repeated evolution via de novo mutation, right? So we go from, in these examples, this would be serious repeated evolution. And independent origin. So we're not pseudo-replicating when we're talking about what we can learn about evolutionary biology. But this gene flow problem is a major problem for those kinds of questions. Like, does evolution work the same way every single time? Well, if I'm pseudo-replicating it, I'm not getting a very good picture of that. And so what I've found is a population that doesn't have this gene flow issue. And so we've been able to remove that aspect. And so this population is located in Texas, and it's a collaboration with Andy Glusenkamp at the San Antonio Zoo. And so we had a reviewer that was trying to say, well, how do you know it's not gene flow? And it's actually that this is 600 kilometers north of the other population. So I feel pretty confident that a little fish hasn't been swimming up 600 kilometers through the desert, but who knows, it could be. And so what has happened is that Osteonyx was brought from the Rio Grande to this region and thrown into the springs as bait fish. And they exploded in their population size. So I'm just going to show you kind of these are the museum records of being able to find Osteonyx mexicanus in Texas. And about 1950, we just see this like massive number of populations spring up. And sure enough, after a hurricane, or it was a tropical storm in 1978, they started to find fish in this cave. And prior to that tropical storm, they didn't find fish in that cave. So we can place this probably at about a 40-year-old cave population. And so this is a collaboration with many different people. And what we're finding with this population is that the cave fish in Texas are starting to look like the cave fish in Mexico. So we have this example where we have lighter cave fish than surface fish. Also, the number of neuromass. So we're going to look at the tiny dots, not the canal. These are canal neuromass, and these are superficial neuromass. The little dots are superficial neuromass. We're looking at those. Those have more in the cave than the surface, and that's also similar to the Mexican cavefish. We can also starve them, and like cavefish can be starved, and they're just not as hungry after a 5-day starve as surface fish are. The surface fish are ravenous, but cavefish just aren't as hungry. And then they also show this wall following behavior. So we put them in a bucket and we mimic, darkness for them. And in cavefish in Mexico, they do wall following because it helps them, figure out where they are in space. And we see the same behavior in caves in Texas versus surface fish. They're just all over the place. So this would be one little fish that's going through, and the red is where it was mostly. We're really excited about this population, and we've been doing more work in this population. We found 2 other caves in Texas as well that look like they're evolving towards a cave phenotype. We've taken these guys in the lab, and we're breeding them right now to make sure that these phenotypes hold steady and that it's actually genetically based change and it's not a plasticity. So, so with that, I wanted to briefly touch on some new work that we've been doing, and that's in the evolution of sleep loss. And so for this, we look at these, so cavefish have less sleep than surface fish do, but I always get this question of like, how do you know that they're sleeping? They don't have eyes, they can't close their eyes. Also, you know, can you put electrodes on cavefish heads? And these things we can't do, but we use a behavioral definition of sleep. And for this, I look for these 4 different characteristics. The first is this prolonged behavioral quiescence, often accompanied by preferred location or characteristic posture. There's also a decreased responsiveness to their surroundings and a rapid reversibility. So this distinguishes sleep from coma in that you can wake someone up pretty easily. Lastly, it's under homeostatic control. So this means that there's like, if you lose your episodes of sleep, you can think of this last time you took a really long haul flight, or you stayed up all night cramming for a test, that you need to sleep later. You have to rebound from that. You have homeostatic pressure to make up that sleep. And what's remarkable is that sleep predates like the earliest branches of evolution in animals. So even Cassiopeia, the upside down jellyfish, enters this nightly period of quiescence in which their rhythmic pulsing actually slows down, and they exhibit these 4 characteristics of sleep. If you keep them from exhibiting this quiescent period, they have to make it up. So they actually have rebound. Sleep is one of those really interesting problems in evolutionary biology that's largely been ignored, save for a few labs. And it's that, you know, there's such diversity across metazoans in the amount of sleep and the sleep phenotypes. So you have this like big brown bat that will sleep for 20 hours per day, and then elephants can get by with only 2 hours a day. Sometimes elephants can go days without sleeping. And there's barely any generalizations that we can make. The one thing that does seem to be holding steady is that carnivores sleep more than omnivores, and omnivores sleep more than herbivores. So it seems like if you have a very nutrient-rich, nutrient-dense food source, that you're able to spend more of your time in a sleeping state. So that's also the case for cavefish, in that cavefish seem to sleep less than surface fish do. So this is a figure from Dubuis et al. 2011. And what they've shown is that in the populations I've been talking about, Pichon, Tanaha, and Molino, the surface fish will sleep a normal amount and the cavefish sleep less, both in the daytime and at night, meaning that overall they sleep less totally than surface fish do. So what we found is that, you know, in short sleepers in humans, right, we have this example where you have sleep loss correlating with increased appetite, insulin insensitivity. Short sleeping individuals are more likely to develop obesity and type 2 diabetes, and that's exactly what we see in cavefish. So the sleep duration in cavefish is less, right? They eat much more. They have hyperphagia. So they consume more food, and they also have higher fat per protein body content. What we think is happening here is that so what we think is happening here is it's all food dependent. These traits are food dependent, and that's if you take a surface fish and you're feeding them normally, and then you starve them for 30 days, they will actually reduce their sleep similar to cavefish. Alternatively, if you take cavefish and you starve them for 30 days, they increase their sleep. And so what we think is happening is that in the lab, where we have to keep them fed every day to make our animal care people happy, they're in the state of wet season. So in the wet season, there can be nutrients being brought into the cave systems, and the cavefish, we think, the hypothesis is that they stay up and they eat and they eat and they eat, and they pack on the milligrams for those lean times in the dry season. So we think by feeding them all the time in the lab, we're basically inducing a state of sleeplessness, whereas during the dry season, they actually quiesce down, and the exact opposite happens in surface fish. So we have a couple grants with a great group of people to be able to start to look at these things. And one of the first things we wanted to know was whether or not they, while they have lower baseline sleep, do they also have sleep homeostasis that remains intact? And so what we wanted test then was what happens when we sleep deprive them. So to test this, we took a clutch of, multiple clutches of surface and multiple clutches of cave. We did 3 different cave populations, and we sleep deprived them overnight for 10 hours. And we flash froze them right at 6 a.m., so after they've been sleep deprived. And this is my postdoc, Courtney Passot. And what we have rigged up here is that they go into these bottles and they get shaken every minute randomly so that they cannot sleep throughout that 10-hour period. And we know that this causes rebound because we see it in the behavioral data. So, so here is an example where we are acclimating them, we sleep deprive them, and then they have some recovery time. And in the behavioral data, what we see is that, the surface fish definitely rebound. So this is total sleep in the control, and then in the sleep deprived, they rebound after sleep deprivation. We see similar rebounds in the cavefish. This goes away largely by the time they reach tonight. So they basically make a rebound. They have a homeostatic impulse to sleep. They make up the sleep, and then they don't need to do that too much longer. We see this also in the genetic data. So we did RNA-seq on whole 30-day individuals. And what I was hoping to see is that we would see lots of stress and lots of other types of genes being completely dysregulated in surface fish among sleep deprivation, and the cavefish being largely robust in their gene expression. And that's not really what we see. We see pretty much equal responses gene expression-wise between cave and surface fish. So I'm going to conclude today by saying one more kind of public service announcement, right? One more cautionary tale. And that's that when we were getting ready to do the gene expression work for this, my postdoc and I were having a conversation, and she said, I think we can just, put them in RNA later and call it good, and we'll do the extractions based off the RNA later stored samples. And I've been like really skeptical of RNA later. I was like, well, how about we do an experiment where you're going to take a clutch of individuals, and we're going to flash freeze 6 of them at 6 a.m. And we're going to take another 5 individuals and put them in RNA later. And we'll leave them on the benchtop for 2.5 weeks. And so this was to mimic exactly what she had done when she was doing fieldwork, where she did not have reasonable refrigeration. And so she just had to leave them out for 2 weeks till she could get them back on the bench. And so what she showed, and so then she did the RNA-seq on these guys. So what we showed is that while sleep deprivation experiment accounted for maybe this experiment accounted for 12 to 16% of the variation in the data, storage condition, so this would be the RNA later in red and the liquid nitrogen preserved in blue, storage condition alone accounts for 27% of the variation explained in the data. And so this particular graph, keep in mind the rates here. What I'm showing you in this graph, these are the genes that are differentially expressed only to RNA later. This is that graph I just showed you before. So what you can see is that by and large, RNA later is dwarfing any signals of biology that you can pick up here. More so, what's crazier is that it does so in a non-random way. So genes with higher mean expression, genes with more exons, genes with less GC, are all associated with higher abundance in RNA later treatments. So it's a non-random, it's very much a biased process. In addition, we can take those things that are upregulated at the RNA later and treatment and look at an enrichment analysis, and that's the only time when we actually see enriched for certain categories for an actual significant enrichment. And sure enough, those guys are enriched for RNA processing, functional categories dealing with RNA processing. So we think that the RNA later might actually be altering the physiology of the sample. So the take-home message for this part of the talk is that indeed sleep homeostasis is intact in the cavefish. So despite this reduced overall baseline sleep duration, they still have this need to sleep, even if it's less than what surface fish sleep. And that storage condition can completely dwarf biological signal. So with that, I'd like to thank the osteonics community, and again, thank Patty for all of her help and being such a great collaborator. And Marco came to the lab for a while and really enjoyed his time in the lab as well. And then as well some funding sources and some people that have been in the lab. And then following that, I'll just take questions.
Thank you very much, Susan, for your great talk. We have several questions actually by the audience, so I'm happy to share them with you. The first one is, the two lineages of cavefish could be considered as new species?
Well, potentially. And so there's some data from Richard Borarski where he can cross between the two lineages. And actually, it's really when he does a surface, and I think it's a new lineage surface to an old lineage cave. And sorry, I should say it a different way. It's like the pink lineage surface to the blue lineage cave. If he crosses those two, in the hybrid males, they have some kind of sterility, I think.
Yeah.
Patty, you remember correctly what was happening there?
Yeah, he was maybe telling something like there was a very reduced fecundity between these two crosses.
Yeah. And it was always old to new or red to blue, you know? So, yeah.
Yeah. So the next question is from Hernan Vasquez. It's more related with the RNAlator procedure. So he's asking, RNAlator only preserves RNA at room temperature for less than 72 hours. It's not surprising to see those huge gene expression differences.
Yeah.
RNAlator stored samples need to be kept at least at minus 20 degrees.
Yeah. So, yeah, and that's absolutely true. But what we wanted to do in this example was that, like, actually mimic what my previous postdoc had done in the field. And she just didn't have an option because she was in a really remote location. And so she didn't have an option to be able to stick them in the minus 20. And so we wanted to get an idea of how much that could impact, in real field conditions. And by and large, when you look at RNAlator papers, the majority of people use it correctly, where they, and we would, funds weren't available to be able to take it, do a benchtop one, a 4-degree one, a negative 20 degree one, and do it tiered like that. But but we did at least have a cautionary tale for what can happen whenever you leave it, in a room temperature environment.
Yeah, that is for sure. Yeah. So in my case, for example, I must admit that sometimes you don't have access to this kind of procedures in the field. So it's totally worth it to know that there is a bias in the procedure.
But I think the answer is, like, if you can at least get them on ice, it's better than anything,
Yeah, that is true. So the next one is from Marco.
Oh, hi, Marco.
So I don't know. Do you think this standing genetic variation in surface fishes possibly plays a role in terms of how quick can be achieved cave-dwelling phenotypes in Astyanax?
Yeah, I think so. I mean, that's an excellent question, and something we are looking at right now with another population in collaboration with you guys, right? With Chica, and so we're seeing that we're seeing differential retention of surface alleles, and the same thing is happening in another population too that Nick Rohner is looking in, and so I think we're just beginning to try to understand that, and it's a really cool question for sure, because I think we do see a low level of cave alleles floating around on the surface, and maybe those could catalyze. I think what would be incredibly interesting, and something we're trying to tease apart right now, but it's difficult, is to try to look for things like supergenes or inversions that could easily find their way, even if they're in a heterozygous state, if they find their way into a cave population and they have everything locked up that's important for cave adaptation, that they can take off once they go into a cave population. So we're looking at, we have some whole genome sequences from a lot of different cave populations, and we're looking at that, largely due to to you guys sending us flash frozen samples. So
yeah,
they were worth it. They really were.
It will be. So again, from Hernan Vasquez, did you find evidence of male and female bias gene flow in Astyanax?
Oh, that's a great question, and I don't think we looked at that. So, no, we haven't looked at that. But however, there are. Some of Patty's work has shown that some of these populations have mitochondrial DNA from one lineage and surface DNA nuclear DNA from another lineage, and so there's definitely something going on there.
Yeah, that is very interesting. So I have a question, if there is not others by the public. So it's more related with the demography, Suzanne. What do you, when I was checking these Molino graphs related with the fixation of some alleles, what do you think about the sample size? Because we have found very, very different caves that could be really related with the number of fishes inside of it. So do you think that could be very different, the scenarios, depending on the caves?
Absolutely, yeah. I mean, yeah, do you mean like the time since divergence, or do you mean?
Yeah, time since divergence.
Yeah, and I think that now that we have this larger data set, we can do the modeling on that and be able to, but it's really complicated by the gene flow, right? Because, and that, you know, daddy's good because it does account for gene flow, but a lot of times that can obscure your estimates of how early in time, or how recently they diverged. So yeah.
Super. We have another question. So it's Ulises Rosa. Hi. When cavefish sleep, what environment clues do they use in order to know they should be sleeping, and when they should be awake?
Yeah, that's a great question. I don't think we know. We have not. So in the lab, usually these studies are done on they're usually done with a light cycle, because we want to keep it consistent between surface and cave. And they do have an active pineal gland, so they are able to detect the light. And we don't know what they're doing in the cave. We have never videoed them sleeping in the cave. And we had a really intense field expedition planned in March, where we were all supposed to go out and actually be able to get a picture of what's happening in the cave environment, and that had to be delayed because of COVID. But in January, I'll be going down to Texas, and we'll be trying to do a little bit of that in Texas, at least, to try to get some data on that.
So the second part of the question, how their circadian rhythms became trained.
Yeah, and so one thing I didn't have a chance to talk about is that it's up for debate on how active how good their circadian rhythm is. And so I can show you, so this is another gene expression study that we did. And so there's about 539 genes that are rhythmic in the surface populations, and at least one cave population for 70, almost 80% of those genes. At least one of the cave populations is not rhythmic for that gene. There's 22% of these original 539 genes, at least 22% of those are not rhythmic in all three caves. And so it's pretty disrupted, the circadian rhythm. And you can see that best when you look at the heat map going from like 0 to 24 hours over a 24-hour period. So this would be a gene that's like really highly active, right, in the morning. And then it tapers off in the afternoon, and then it gets back up to being active. And when we look at our cave populations, they're all messed up. So like you can kind of squint a little bit and see it, but not really well. And these two are more closely related to one another, and you can almost see that in the patterning as well. So these are the 539 genes that you're looking at from the surface fish, and you can really tell that their circadian rhythms are substantially impaired.
Wow.
Yeah. So anyway, yeah, thanks for the question. That's a good one.
Yeah. Thank you very much, Suzanne. We don't have any further questions, but
thank you. Yeah, this was excellent. I so much enjoyed it. I really appreciate the opportunity to speak. Thank you so much.
Thank you.
Thank you.
Bye-bye.
Bueno, quiero nada más avisarle a todos que la próxima semana vamos a tener 2 seminarios, y el del martes a las 11 con el doctor Francisco Vergara Silva, y el jueves 12 con la doctora Irma Gavilán. Por favor, suscríbanse al canal y apachurren la campanita para recibir las notificaciones y que no se pierdan ninguno de los seminarios. Muchas gracias a todos.
Gracias, hasta luego.
Hasta luego.